Hi, I'm Cheyenne Lupton, and this is my second video for Exam 3. This is on the Nirenberg and Matthaei and letter experiments. Translation is a part of writing the genetic code. It is when the nucleotides A, U, C, and G are translated into amino acids. The nucleotides are read in groups of 3, or triplets, and they're called codons. Each codon codes for a specific protein. Did you ever wonder how we figured out which codons are for what protein? Well, Nirenberg, Matthaei, and letter set out to figure it out. The Nirenberg and Matthaei experiment started by creating an mRNA molecule of only uracils, called a polyuracil. They added it to a cell-free extract with all 20 amino acids. One amino acid was labeled radioactive while the other 19 were not. The tube was incubated and translation took place. If the resulting protein was radioactive, that means the codon codes for that amino acid. This was done for each of the amino acids being radioactive in the original tube, meaning 20 experiments were done. The researchers ended up finding that the polyuracil always coded for phenylalanine, which means that UUU will always be that. They repeated this experiment with the other polynucleotides and found that AAA codes for proline, CCC codes for lysine, and GGG doesn't code for anything. This was the start of cracking the genetic code. In the next experiment, done by Nirenberg and letter, short mRNA molecules that had every possible codon were used with ribosomes and tRNAs attached to amino acids. Like before, one amino acid was radioactive while the rest were not. When doing this experiment, however, the scientists let the codon bind to the tRNA. If all of the binding occurs to the radioactive amino acid, the molecule will be caught by some filter paper, and that means the codon codes for that amino acid. This method was used to discover all of the rest of our codons. Overall, both of these experiments are very important to the field of genetics, as without them we would have no idea which codons and amino acids match up.